Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled EndoC BH5 Human Beta Cells in T1 and T2 Diabetes Models. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Mercodia, who developed the content for this presentation. Mercodia is a world-leading manufacturer of cardiometabolic immunoassays, using their 30-plus years of experience to provide tailored bioanalysis solutions for pharma and biotech companies. Mercodia's vision is to improve human health by offering their products and services to scientists around the globe. Their team of experts quickly provide reproducibility, reliable, and high-quality data that meets regulatory requirements. They prioritize speed and flexibility while delivering cost-effective solutions and are a trusted partner to some of the most renowned universities, pharmaceutical, and biotech companies worldwide. Now, it's my pleasure to introduce our speakers for today's webinar. And first, I'd like to introduce you to Dr. Bruno Blanquet. And there he is. Hi, Bruno. Chief Scientific Officer at Human Cell Design. Bruno is currently responsible for the R&D strategy and development of new products, models, and solutions at Human Cell Design. Bruno holds a PhD in immunology and has an extensive experience in cellular systems and control of cell differentiation and specification with over 20 years of experience. Bruno also has extensive expertise in model and assay development, including various tools and techniques. Bruno, together with the Human Cell Design team, are experts in developing physiological and pharmacologically predictive functional human cellular models, assays, and services. And now it's my pleasure to introduce you to your second speaker, and he is Dr. Miguel Sainz, and there he is. Hello, Miguel. He is a scientific affairs manager at Mercodia. Miguel has a PhD in biomedical research and vast experience in tumor and vascular biology, with over 15 years of scientific experience in translational and basic research. Miguel is responsible for the development of scientific strategies for Mercodia in alignment with the company's global view, building and maintaining scientific and medical partnerships. He has worked with multiple experimental models in vitro and in vivo and evaluation methods for protein analysis and signaling. Now, with the introductions done, it's now my pleasure to pass the mic and the controls over to our first speaker, Bruno. So Bruno, when you're ready, you may begin.
Hi, thank you, Sonia, for the introduction, and thank you all for attending this webinar about the use of human primary-derived EndoC BH5 cells in diabetes modeling. And thank you, of course, also to Mercodia for organizing this session and giving us the opportunity to share about the tool that we have developed and the services that we offer and that we can develop for you. I'll start with a brief introduction about our company, Human Cell Design, formerly named Universal Biosolution. Human Cell Design is a preclinical contract research organization, as well as a product development and sales company. We are driven by innovation, which is highlighted by the development of the quite unique EndoC BH human pancreatic beta cell family. We have more than 15 years of expertise in human cell model and assay development. And our services, assays, and products have been used by more than 200 laboratories worldwide. and generating more than 150 publications to date. In order to reach the highest standards of service and products, our approach will combine an industrial approach of standardized and highly controlled model development and production with highly innovative human primary cell-derived models, which, because of their human origin and close to native functionality, give rise to translational tools. Our objective is to be able to support fundamental and drug development research from early target discovery through compound selection to candidate drug optimization and mechanism of action studies. And we think that this is possible because of translational assays that are based on human cell models and validated reproducibility over batches and over the years. The assays that are currently available for type 1 and type 2 diabetes include T-cell mediated beta cell specific killing, as seen in type 1 diabetes, cytokine mediated beta cell inflammation, free fatty acid and glucose mediated glucolipotoxicity, as well as study of GLP-1 and GIP receptor signaling. This is completed by customized services for disease modeling, target identification, and assay development. I will later describe the type of data that can be obtained and questions that can be asked using these different assays. Also of importance, these assays utilize several human pancreatic beta cell models that are also available for implementation in your laboratories. Sorry. So healthy beta cell models include broadly used EndoC BH1 cells as well as EndoC BH3 cells. And these models are using optimized, highly differentiated, mature, and functional EndoC BH5 cells and their derivatives, such as HLA-A2 expressing EndoC BH5 cells and glucolipotoxicity sensitive EndoC BH5 cells. I'm now going to discuss each assay in more details, and I will start by describing EndoC BH5 cells. They are human pancreatic beta cells. They were derived from primary human beta cells through targeted immortalization and initial in vivo derivation and selection. In the production process, they undergo in vitro functional maturation, which involves their de-immortalization. They then follow a process of validation that includes functional validation and pharmacological validation in order to generate highly reproducible batches of cells. In terms of identity, EndoC BH5 cells are homogeneous insulin positive human pancreatic beta cells. They also homogeneously express beta cell specific transcription factors, PDX1 and NKX6.1, seen here in flow cytometry results, and also here by immunofluorescence for PDX1 co-labeling with insulin. In terms of insulin secretion function, EndoC BH5 reproducibly respond and reproducibly, not only reproducibly, but also dose-dependently respond to glucose. And the highest increase in insulin secretion happens between 5.5 and 8 millimolar glucose, similar to what you would expect from native human beta cells. These cells also reproducibly and dose-dependently respond to GLP-1 receptor agonists, and here exendin 4 and semaglutide in red. And they do respond as well to GIP receptor agonists here, also in a dose-dependent manner. Finally, their responses are extremely reproducible from batch to batch. These box plots show all the batches of EndoC BH5 cells that were available at the time the figure was generated. And it shows the distribution of glucose here and glucose plus exendin responses for all these batches. And you can see that the results are very reproducible in terms of magnitude of response, but also in terms of absolute values of secretion. As a conclusion, the use of EndoC BH5 cells guarantee access to primary-like human beta cells that also guarantee reproducible insulin secretion function and reproducible response to incretins. Now, how do we go from healthy in vitro human beta cells to disease modeling and type 1 and type 2 diabetes assays? For that, we developed additional cellular systems. We developed protocols that allow to place wild-type EndoC BH5 cells in disease culture conditions, such as cytokine treatment that induces their inflammation. We developed HLA-A2 expressing EndoC BH5 cells that allow to study T-cell mediated killing using HLA-A2 restricted CD8 T-cell clones. And we developed EndoC BH5 cells that are sensitive to high glucose and lipids to model glucolipotoxicity. Doing that, we generated a set of disease models which are highly relevant both in terms of assay development and as physiological model and thus relevant for translational studies. Relevance in assay development comes from the fact that the reproducibility of this cellular model is well established, and also because they are available for large-scale studies. In terms of physiological relevance, on the other hand, this comes from the fact that these cells are highly comparable to primary beta cells in terms of identity, but also in terms of function. These cells spontaneously express beta cell receptors and signaling pathways, such as GLP-1 and GIP signaling pathways, which are common targets for pharmacology. And they model disease mechanisms in a human and controlled environment, which makes them translational tools. I will now describe in more details each disease model and assay. And for that, I will start with a CD8 T lymphocyte mediated beta cell killing assay. This assay, the objective of this assay is to model the central mechanism of beta cell loss in autoimmune type 1 diabetes. The assay starts, and it will be the case of all of our assays, with thawing of the beta cells. This because these cells are stored as ready-to-use and functionally validated material that can be used at any time. In this particular assay, we use HLA-A2 expressing EndoC BH5 cells as targets of CD8 T cells, whereas wild-type EndoC BH5 cells are used as internal negative control. After a few days of pre-culture, cells are deattached and differentially labeled before to be mixed at a 1-to-1 ratio. This mixed beta cell culture is then co-culture with pre-pro-insulin specific HLA-A2 restricted human CD8 T cells. And after 6 hours, relative survival of each beta cell is measured by flow cytometry in order to quantify HLA-A2 EndoC BH5 beta cell killing. In addition to beta cell killing, various readouts can be measured. T cell activation can be measured as a readout of beta cell T cell immune interaction. HLA expression by beta cells can be measured as a readout of the effect of an inflammatory environment. And finally, insulin secretion, as well as expression of different markers, can be evaluated in order to study mechanisms. This first slide shows a typical result of the measure of HLA-A2 EndoC BH5 beta cell killing. When co-culture with pre-pro-insulin 1524 CD8 T cells, survival of HLA-A2 EndoC BH5 cells is reduced, here 17%, compared to the control cells that represent at the end of this assay 83%. Whereas it's not the case in the presence of irrelevant HLA-A2 restricted CD8 T cells that do not kill HLA-A2 EndoC BH5 cells. So this figure demonstrates the specific killing of HLA-A2 EndoC BH5 cells by pre-proinsulin 1524 CD8 T cells in our assay. When plotted, these flow cytometry results allow to generate curves and histograms showing specific lysis of HLA-A2 EndoC BH5 cells at different effector target ratio. Overall, this data demonstrates the ability of this assay to perform reproducible and quantitative measurement of T-cell mediated beta cell killing. And it just opened the possibility to assess pharmacological intervention as well as mechanisms. In addition, it is important to note that HLA-A2 EndoC BH5 cells are fully functional insulin-secreting cells. Here you can see their response, you can see their insulin secretion in response to glucose and hexandin 4 for HLA-A2 expressing cells compared to their wild-type counterparts. The fact that HLA-A2 EndoC BH5 cells maintain their insulin secretion opened the possibility to measure defects in insulin secretion that could accompany initial immune reaction against these cells. Then, in order to make our autoimmune diabetes assays even more physiological and relevant, it was important for us to integrate the pro-inflammatory environment aspect. In this slide, we show that cytokine treatment increases HLA-A2 expression at the surface of HLA-A2 EndoC BH5 cells, and this is confirmed when measuring fluorescence intensity. It is then possible to ask how inflammation or whether inflammation influences the interaction between T cells and beta cells. For this particular assay, we switched to a different readout. We measured the activation of CD8 T cells when they encounter their target cells. This was done through the measurement of a secreted chemokine, MIP1 beta. And in this assay, HLA-A2 EndoC BH5 cells, but not wild-type EndoC BH5 cells, were able to induce T cell activation here in blue. Then we looked at pre-treatment of HLA EndoC BH5 cells with gamma interferon and IL-1 beta, which mimic an inflammatory environment. And we observed that this treatment potentiates T cell activation by HLA-A2 EndoC BH5 cells, here in red. And finally, we addressed the question of pharmacological intervention. We use baricitinib, which in purple here and here, which is a JAK1/2 inhibitor and a blocker of gamma interferon signaling, and also a clinically approved drug in rheumatoid arthritis. And we observed that baricitinib is able to prevent the potentiation of T cell activation by cytokines. This is also shown here with an histogram with decreased T cell activation in presence of baricitinib compared to the cytokine-treated condition. To conclude on this group of assays that address the question of autoimmune diabetes, we can say that they are clearly useful for a variety of approaches. They can be used to develop beta cell protection strategies. They can be used to develop T cell inhibition strategies. They can also be used as an integrated model to study beta cell T cell interaction in an inflammatory environment. And finally, they can be used to dissect mechanism of action as well as in screening approaches. The advantages of these assays are their physiological relevance with the use of CD8 T cells that are specific for an endogenous beta cell peptide. Another aspect of their physiological relevance is the possibility to measure the consequence of T cell attack on beta cell insulin secretion function. And finally, other advantages include the demonstrated pharmacological validation in the T cell activation assay using baricitinib, and the availability of an internal negative control with wild-type EndoC BH5 cells. The next assay that I will describe is a cytokine-induced beta cell inflammation assay. This assay models the impact of inflammation on beta cells, a phenomenon that occurs in both type 1 and type 2 diabetes. In this assay, EndoC BH5 cells are treated with gamma interferon for a few days, and beta cell death is then measured. Additional readouts that can be measured are insulin secretion, as well as gene expression or expression of specific markers. In addition, in projects looking for therapeutic strategies, test compounds can be applied during the gamma interferon treatment. In this slide, in this first slide, we look at beta cell survival. In this model, gamma interferon induces reproducible EndoC BH5 cell death. In addition, and very importantly for the relevance of the assay in drug development projects, this cytokine-induced beta cell death here is prevented by parallel treatment with JAK1 inhibitor baricitinib. We then went a bit further in terms of disease-related phenotypes and looked at insulin content and secretion. In this model, inflammatory cytokines impair insulin content here, which is restored by baricitinib. We then measured insulin secretion, and gamma interferon treatment impairs glucose-stimulated insulin secretion compared to control cells. And this effect is also prevented by baricitinib. Together, this data demonstrates that our beta cell inflammation model recapitulates some of the hallmarks of inflammation in diabetes with insulin secretion defects and beta cell loss. So we conclude that this assay can now be used to develop beta cell protection approaches to study mechanisms function related to beta cell inflammation. It can also be used to measure and compare compound activities on beta cell inflammation and to develop screening approaches. Here, the advantages of this assay are its physiological relevance with inflammation-mediated beta cell loss that is associated with insulin secretion defects. And a second advantage is the already demonstrated pharmacological validation with baricitinib. The next assay that I will be talking about is a glucolipotoxicity-induced beta cell dysfunction assay. This assay models the impact of high free fatty acids and glucose on beta cells, a phenomenon that occurs in type 2 diabetes. In this assay, EndoC BH5 cells, or the modified version of EndoC BH5 cells, that is sensitive to glucolipotoxicity, these cells are treated with high glucose and free fatty acids for a few days, and beta cell death, as well as other readouts, are then measured. As I said, additional readouts exist, and one of them is the measure of insulin secretion, as well as the measure of expressions of markers of identity or functional markers. In addition, and similar to the previous inflammation assay, pharmacological compounds can be assessed in this assay. In this first slide, we look at beta cell survival. And in this assay, high glucose and palmitate treatment induces reproducible EndoC BH5 cell death here. Importantly, this glucolipotoxicity-induced beta cell death here is partially prevented by parallel treatment with GLP-1 receptor agonist exendin 4. Now, we also looked at, we also set up the condition to look at insulin secretion in this model, and we found that high glucose and palmitate treated EndoC BH5 cells also present defects in insulin content and insulin secretion. In the first figure, you can see that glucolipotoxicity impairs insulin content, or glucolipotoxic condition, sorry, impairs insulin content of these cells. And we then measured insulin secretion, and glucolipotoxic conditions also impaired insulin secretion here compared to non-treated cells. And this is seen both in terms of basal secretion that is affected, but also in terms of glucose and exendin 4 stimulated secretion that are decreased compared to control condition. As a conclusion on this part, this assay recapitulates some mechanisms of glucolipotoxicity in type 2 diabetes with insulin secretion defects and eventual beta cell loss. It can too be used to develop beta cell protection approaches to study mechanisms of action related to glucolipotoxicity. It can also be used to measure and compare compound activities on glucolipotoxicity and to develop screening approaches. And the advantages of this assay are, again, its physiological relevance with glucolipotoxicity mediated beta cell loss that is associated with insulin secretion defects. And the second advantage is the already demonstrated pharmacological validation with GLP-1 receptor agonist exendin 4 able to prevent some of the defect observed, and that can be used as control in future studies. Another type of assays that is made possible with our human beta cell models is assays related to GLP-1 receptor and GIP receptor signaling and the screening of insulin secretagogues. In this assay, EndoC BH5 cells are treated with test and reference compounds, and insulin secretion is measured. Importantly, these assays can be performed right after thawing, as well as after up to 4 weeks of culture, allowing to combine to disease modeling or to treat with other compounds. In addition to insulin secretion, cyclic AMP production can be used as a readout of secretagogue activity. And finally, other readouts such as gene expression can be used to address the question of mechanisms. This slide will show data that I've already shown before, but I need to repeat them here in order to introduce the following slide. It shows that EndoC BH5 cells does dependently respond to GLP-1 receptor agonist, exendin 4 and semaglutide, which in this case potentiate glucose-stimulated insulin secretion. And the second figure is that EndoC BH5 cells also does dependently respond to GIP receptor agonist, which also potentiate glucose-stimulated insulin secretion. Because of these highly reproducible dose-dependent responses, EndoC BH5 cells can now be used to rank compounds. And this not only in terms of EC50s, but also in terms of maximum efficacy. And here we show the example of 2 compounds that were compared to exendin 4 and semaglutide. Finally, in this assay, it is possible to verify the specificity of the compound using antagonist. And here is an example with the inhibition of exendin 4 activity using GLP-1 receptor antagonist exendin 9. To conclude on this assay, this secretagogue assay using EndoC BH5 cells, it can be used to measure secretagogue activities. It can be used to rank compounds. It can be used in a screening campaign, as well as to study mechanisms of action. And the advantages of these assays are a physiological relevance due to the fact that the cells endogenously express functional GLP-1 receptor and GIP receptor signaling pathway. Another advantage is the fact that the high sensibility and reproducibility that we observe is absolutely essential for ranking and screening approach, and we do have this reproducibility with these cells. Other advantages then are the pharmacological validation that already exists with reference molecules on these cells, and the possibility to run chronic studies with up to 4 weeks maintenance of these cells in culture, as well as large studies using small well plates. I'm now switching to the last model I will be discussing today, and it is the use of EndoC BH5 cells in target validation approaches. EndoC BH5 cells can be transfected or transduced in order to express siRNA, shRNA, microRNA transgenes. And this in order to identify targets or actors of disease mechanisms of or drug effects. Sorry. In addition, there is two types of approaches that can be developed: a transient approach for rapid proof of concept, and a customized stable knockdown or transient approach that we can develop in order to provide ready-to-use functionally validated material. In terms of readouts, different readouts can be used following this type of approach: beta cell death or survival, insulin secretion function, as well as looking at the expression of functional marker or whole genome expression profiling. In this slide is presented an example of a transient target validation approach for rapid proof of concept. Our hypothesis here was that knockdown of this particular gene would result in increased sensitivity to glucolipotoxicity. In this first figure, we determined that efficient knockdown of the target gene can be achieved. We then looked at cell survival in glucolipotoxic conditions. Non-transfected cells and control siRNA transfected cells are not affected by palmitate and high glucose treatment, here with 0.5 millimolar glucose and here with 1.5 millimolar glucose, palmitate, sorry. In contrast, target gene knockdown cells show a decreased survival when challenged with high glucose and palmitate, confirming the initial hypothesis. The same type of proof of concept approach was used by other than us, and I'm giving you an example here. siRNA knockdown for insulin or HNF4 here resulted in their hands in modified insulin secretion compared to control cells. They observed modified insulin secretion in terms of basal secretion in absence of glucose here compared to control cells, as well as stimulated secretion with the two type of knockdowns where the insulin secretion was modified compared to control cells. And this was published by the Novo Nordisk Research Center at Oxford in an effort to develop a platform for target identification. Finally, this slide presents the translation of a proof of concept into the production of a stable customized assay. Following the transient approach that demonstrated sensitivity to glucolipotoxicity in siRNA knockdown cells that I just presented before, the same gene was targeted through viral transduction and stable knockdown. The cells that were produced display reproducible knockdown. And when their sensitivity to glucolipotoxicity was assessed, they did show reproducible sensitivity to palmitate and high glucose treatment in terms of survival. And finally, also in terms of insulin secretion, these figures are the same as the one I presented to exemplify our glucolipotoxicity assay. Just to show you that we can transfer transient experiment into a stable knockdown or overexpressing transient cell line that can give rise to a stable cell product with reproducible phenotypes. So here, the stable knockdown cell, similar to the transient knockdown cell, show decreased insulin secretion after being challenged with palmitate and high glucose. To conclude on these transient and stable modification systems, they have many possible applications. They can be used in target identification and target validation approaches. They can be used for assay development, and they can be used to generate disease models. The advantages of these systems are the possibility of a rapid transient approach that provide rapid proof of concepts. The second advantage is a possibility of a customized stable approach that is available from us that can support long-term projects with ready-to-use validated material. In addition, the generated assays are physiologically relevant with the possibility to combine with analysis of insulin secretion function. And finally, other advantages are the numerous available readouts and the possibility to use these systems in combination with existing disease models. Now, as a conclusion, I will say that human cell design can be a dedicated partner on many aspects of your projects. We can facilitate your transition from preclinical work to clinical studies. This because of reproducible human data together with the already documented physiologically relevant assays. We provide access to integrative human models that allow to address multiple disease-related readouts in parallel, including those related to beta cell function with insulin secretion. And finally, our assays allow to secure long-term scientific strategies because of minimal batch-to-batch variation and proven reproducibility over the years. I'd like to finish by acknowledging the initiators of the EndoC vH cell project, Dr. Schaffman, Dr. Ravasa, and Professor Chernikov, our collaborators on the different studies, and the entire team at Human Cell Design. Thank you for your attention. I'll be happy to answer your questions at the end of the presentation. But right now, we are going to have a poll question. Sonia.
Okay. Thank you very much, Bruno. I will launch that poll question now. And there it is, everyone. This poll question is done in real time, so your participation is strongly encouraged and very much appreciated. So please go ahead and select from the below which ones of the discussed models attracted your interest for potential use. So you have 4 options below to choose from. So please go ahead and make your selection. And it looks like we have half of our audience have participated in our first poll question. So if I can encourage the rest of you to go ahead and launch your poll selection right now, that'd be greatly appreciated. Okay, that looks like it's everyone. So I'm now going to close the polls and let's take a look at the results. Okay, so here are the results. In our audience, 34% of our audience chose CD8T cell cytotoxicity, while 48% says cytokine inflammation. 39% of our audience had glucolipotoxicity, while 27% chose target identification, and then we have 32% of our audience choosing GLP-1R GIPR assay. So thank you everyone for participating in our first poll question. And now it's my pleasure to pass the mic and the controls over to Miguel. So Miguel, when you're ready, you may begin.
So thank you very much, Sonia. Thank you, Bruno, for starting this webinar, and I hope that everyone is enjoying and learning a lot. Now, what I will talk to you about is about the compatibility that Mercodia has with human cell design. So obviously, there are many different tools, and Bruno has shown quite nice results with insulin, where the human cell design team has actually been using the Mercodia assays. So if we try to think about that, about this, just to give a brief introduction, what is Mercodia? Sorry for that. I assume you should be able to see my presentation. So again, I was going to just talk to you about what is Mercodia. Some of you know that Mercodia is a global immunoassay development leader with more than 30 years of experience, and we actually have our strength in developing tailored bioanalytical services and immunoassays. And some of the things that we can develop for you, of course, it's from the bioanalytical service perspective, where we can develop assays that are fit to purpose or perform the bioanalysis in preclinical and clinical development. We can do custom production of different assays, and of course, considering all the reagents and components that they would be required. And we also have our catalog with products that they're gold standard immunoassays for cardiometabolic diseases. Now, where are we located? Mercodia has the headquarters in Uppsala, Sweden, as hopefully most of you know. We also have laboratories in the US, in Winston-Salem, plus we also have the subsidiaries in France, in Paris, plus multiple other places where you can find us via our distributors. And this is just to show a little bit of how far can we reach, one could say. If we think about Mercodia, we have a strong product portfolio where we are a world-leading provider of immunoassays in the field, as I mentioned before, cardiometabolic diseases, and we have gold standard immunoassays where we have 30 ELISA kits, 1-bit assays, and more to come, and we have 12 supplementary products. And of course, each of them has their own strengths. The intention that we have at Mercodia is to meet the needs in different therapeutic areas. And when I refer to this, I want to emphasize that the assays can be used in different fields, such as in the medical field, biochemistry, molecular biology, or bioengineering, and of course, in the different areas that they are within these fields. However, of course, we will have to consider the biomarkers that our assays work for, which organs are they expressed, but we know also that that could be involved with basic research, preclinical research, or clinical research. Among the assays, and as it was mentioned, I was mentioning at the very beginning, we have the insulin ELISA, and I will mention right now the two insulin ELISAs specific for human samples that we have in our catalog. On one hand, we have the insulin ELISA 1113, and we know that this is a straightforward protocol. It has a straightforward protocol that is composed by ready-to-use components with high specificity, and it's accurately measuring human insulin. So this is one of the advantages of using this product with the cells from Human Cell Design, for example. On the other hand, we have the ultrasensitive insulin ELISA, that it's a kit that comes with a straightforward protocol, again, ready-to-use components, and it's high specificity, and it's actually enables you to accurately measure this human insulin at very low levels. We have as well other insulin tools within our repertoire. So we have, if you have a huge amount of samples, we have the total insulin Northern Lights MBEATS assay, which is a multi-species high throughput tool that allows you to perform the measurements of insulin in both low and high levels of insulin, and also thinking about small sample volumes without or reducing the tedious dilutions that sometimes are required. And of course, depending on the experimental setup, sometimes you might want to use either the ISO insulin ELISA or the LISPRO Northern Light ELISA. Now, if we think about the ISO insulin ELISA, this will help us to accurately measure insulin in most insulin analogs in different species. So depending on the experimental setup, you can combine ISO insulin with the human insulin ELISA. If we think about the LISPRO ELISA, that will allow you to measure specifically the insulin LISPRO without having any cross-reactivity with endogenous insulin or other insulin analogs. Now, what to have in consideration when choosing your right assay? Well, one of the things to think about is the dynamic range. We have insulin assays that will be more specific and more suitable for low values and low concentration of insulin, while we have others that they span in the higher range. And depending again on the experimental setup, you might want to choose some of the other ones like the isoinsulin and LISPRO, depending on the molecules that you're interested in detecting. One of the important things as well to keep in mind is that we don't only have the insulin ELISAs, but we also can think about other assays, other ELISAs in this case, that detect biomarkers that they are secreted by the same beta cells. So in this case, we can point out the C-peptide ELISA or the ultrasensitive C-peptide ELISA. So in this case, of course, reducing or avoiding cross-reactivity with other molecules, and depending again on what is the concentration that it expected. If it's within higher value, you can go for the C-peptide 1136, or if you have low levels of C-peptide, you can use the 1141. Other molecule to have in mind would be proinsulin, and we also have that ELISA here. And this one, for example, would be samples at low levels and with no cross-reactivity to human insulin or C-peptide, which becomes actually quite useful depending on what is our experimental design and what is the setup. Now, we have another poll question here. Sonia.
Thank you very much, Miguel. I will launch our final poll question for our audience members to engage in, and there it is now. And the question is, have you ever used a Mercodia assay with in vitro samples? So please go ahead and make your selection. And since this is a simple question, I will leave the polls open for a few more seconds. It looks like we have the majority of our audience have cast their votes. We have a few other people there who are still making a decision, so I will leave it open for a few more seconds, and then I will close the polls. Okay, that looks like it's everyone, so I'm now going to close the polls and say thank you to everyone for participating in our poll today, our final poll, and here are the results. So in our audience, Miguel, we have 72% that said yes, 21% said no, 0% said I have tried, and then 8% said never thought about it, but I'm interested. So thank you everyone for participating in our final poll question. Now back to you, Miguel, and your thoughts on the results of all the poll question. Anything surprising with them?
Thank you, Sonia. No, I don't think, I mean, I'm not surprised in some of the questions, at least not this last one. Where I think that we will be able to discuss them during the question session at the end. But for now, I would like to thank you all for listening to the presentation. And as Sonia mentioned initially, we will jump after this to the question and answers session. Thank you. And Sonia, up to you.
Okay, well, thank you very much. And with that, we'll put on our webcam. So I'll invite Bruno to put on his webcam. Hi, everyone. Now, remember, you still can continue to send in your questions by using that questions window, and we'll try to answer your questions during the time we have together. So let me start with this first question here. This audience member is asking, can you conduct comparisons between the progression of insulin secretion defects and that of other disease-related phenotypes? Who would like to start with that one?
I think this
Bruno's
question is
smiling.
addressed to me.
Okay.
No, I believe it's addressed to me. Yeah, of course. I mean, that's the idea, and that's why I insisted on the insulin secretion aspect, because it is the main function of these beta cells for sure. Mm-hmm. And a lot of time when we look at disease models, we have to oversimplify. And here, I think with these cells and with the kits from Mercodia, of course, we have a model where we can really look at several disease-related phenotypes in parallel, and some of them are very meaningful. Beta cell death, of course, but also beta cell loss of function. So it's really the idea of these models to allow you to compare different disease-related phenotypes with a highly functional cell model.
Thank you for that. Miguel, anything to add to that before I jump to the next question? Okay, thank you. We have so many questions coming in from the audience here, so that's why I'm busy trying to get everything organized. All right, we'll jump to this question here. Did you say that that you need to differentiate the EndoC BH5 cells? They're looking for clarification.
So I didn't say that, maybe I misexplained. No, okay. The cells are ready, they are functional when you receive them. They have been frozen as functional beta cells. So there is no further differentiation to be done. We take care of the maturation, differentiation. Yeah, so they are used, the assays that you've seen, the cells are taken from the freezer or from nitrogen, in fact, that's very important. They are put into culture, and after just a few hours or days, you can run your assay, and the insulin secretion function, for example, is already here and fully functional. Okay. And sorry for misexplaining it during the webinar.
Well, thank you for clarifying that. I'm sure they appreciate that. I'll jump to the next question now, Bruno and Miguel. This audience member is asking, what is the source of the PPI-15CD8 T cells?
So this work has been done as a collaboration with the lab of Roberto Malone in Paris. So the source is the CD8 T cell that they use. Currently, because I think there is a question that is connected to that about the availability of the cells. So at the moment, the cells are not available per se. What we are setting up is a platform to run the assay ourselves, And in the future, as soon as possible, we would like to have the cells available as well. But it's not the case currently. However, if you do have interest in this assay, please contact us and we'll see how fast we can either develop something for you or with you, or we will keep you informed with how long it will take to have the cells available. So yeah, the source is the group of Roberto Malone in Paris.
Put on my mic. There you go. Thank you. All right, we'll go to the next question here. Is it possible to use the Mercodia assays with cell lysates?
And yeah, here I can, thank you for the question, by the way. And here I can just say that yes, indeed, actually, it has been shown not only by Mercodia but also by multiple researchers that using the cell lysates, it's an accurate way to use the assay, depending on what is the molecule that is desired. And of course, sometimes there might be questions about, like, for example, what kind of buffers have been used to produce the lysate or the storage for the storage of the lysate. And I mean, we have several tech notes that can always help for the different users, so they can confirm that there is no cross-reactivity and that the assay is performing the best way.
Okay. All right, thank you very much for that. We'll go to the next question. This audience member is asking, has tirzepatide being tested on EndoC BH5 cells?
Yes, I haven't shown the data. Well, you've seen that. It has been tested. And as a dual agonist, of GLP-1 receptor and GIP receptor, we did compare to GLP-1 and to GIP. We did see an effect that is not, in terms of maximum efficacy, it's very similar to what we see with either one of the two agonists. What we have started to do is to look at potential, well, whether it goes through GIP receptor or GLP-1 receptor mostly. And we did observe that it seems like there might be a bias toward more function through the GIP receptor. So we were very happy to see that it's actually what is now being described for this molecule. That in fact in the human cells, it might human beta cells at least, the most of the activity of tirzepatide might go through GIP receptor, and it seems like we are observing something similar. The reason why I'm saying that is that we try to block the GLP-1 receptor when treating with tirzepatide, and we saw a limited effect. So right now, it's still under investigation whether what we see is very similar to what is described now in by others in primary cells. But yes, we've tried tirzepatide. It does work, and it does seem to show the same bias as it is recently reported from primary cells.
Okay, thank you so much for that detailed answer there, Bruno. We'll jump to the next question. With the popularity of GLP-1 RA drugs, are there plans for Mercodia's to develop active GLP-1 ELISAs?
So for GLP-1 ELISAs, we already have one ELISA out in the market, if it comes to that. So, and we know by some researchers that they have been using it. We have been in collaboration with some of them also to prove that it's effective. Now, if it's thinking about another kind of specific molecule, if I'm not missing the question, like then I don't think that we are actually right now yet thinking about one specific. There are so many of them, so many different molecules within the pathways, right? But definitely the GLP and the GIP assays, we have them out.
Okay, thank you, Miguel, for that. All right, we'll go to the next question here. How can I confirm cross-reactivity with other molecules expressed within my samples? That's a great question. Who would like to start with that one?
So I can start, I can say. Well, it depends on, of course, what are the molecules that the people that is using the assay is actually wondering about. So sometimes we can have in mind already specific molecules with the ones we know that there might be cross-reactivity. The antibodies might be detecting some fragment from another molecule that could be abundant in our sample. So there is always a way to confirm that, either using the right controls from the experimental setup, or if someone is unsure about which kind of, or what would be the levels of expression of other molecules within the samples, what they can do is also, you can titrate by using the specific molecule. As an example, if I want to see that there is no cross-reactivity with C-peptide in my insulin ELISA or in my insulin assay, one of the possibilities is to just use different concentrations of C-peptide alone and see if it's being captured. And then I will compare it, of course, with my samples. On the other hand, if I know that my sample should not have C-peptide at all, what would be interesting, but who knows? Maybe if there's no if I don't expect C-peptide, then I can just put my negative and my positive controls within my experimental design. But I don't know, Bruno, in your case, I don't know what would be your experience.
Well, I'm sorry, I missed the question actually.
Oh, no problem, no problem. How could I confirm cross-reactivity with other molecules expressed within my samples?
No, I don't have a better answer than what you said, Miguel. Sorry, because that's not something that we address in our models.
Okay, thank you very much, Bruno. All right, we'll run to the next question here as we have about 3 minutes left. This audience member is asking, can EndoC BH5 cells support regenerative medicine programs?
Yeah, okay, so not as a cell that could be used in human because of the way they were developed, at least not at the moment, for sure. But they could certainly support this type of project as a positive control in differentiation programs from iPS cells, for example, mostly. So as a positive control of the beta cell identity and function, I think they would be great controls. And a second way to use them, and I think that this has already started, in fact, it's to use them in animal models as a proof of concept when developing, well, approaches about biomaterial to encapsulate the cells, for example. So they are very good for proof of concept approaches in regenerative medicine approaches, for sure.
Okay, thank you, Bruno. Anything to add to that, Miguel?
Nothing to add.
Okay, that was perfect. Thank you so much. Well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar. And if we couldn't attend to your questions, and there's a lot of them out there, so I'm so sorry about that, but the team at Mercodia will follow up with you after this presentation. And if you have any further questions, please direct them to the email addresses that you see there on your screen and also the website. Okay, so thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues so they may register for the recording here as well. A survey window will be popping up on your screen. Your participation is encouraged as this will help us to improve our further webinars. And get even more out of today's presentation by downloading the supporting materials that we have available for you under the Handouts tab, and we have 2 there for you, the Gold Standard Immunoassays and a Gut Feeling for Hormones. So please go ahead and check that out. Now, finally, please thank our speakers, Bruno and Miguel, for that very insightful presentation and for answering all your questions. So thank you very much, gentlemen. And there they are. Thank you.
Thank you.
We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until next time, I'm Sonia Hunt. Please take care and bye for now. Bye, everyone. Bye, Miguel. Bye, Bruno.
Bye.
Bye.